All right, I'm super excited to welcome Pratyush Tiwary. He is the Millard and Lee Alexander Professor, where he leads the Center for Therapeutic Discovery at the newly established Institute for Health Computing. Pratyush also received his degrees from IIT BHU and Caltech and completed postdoctoral work at ETH Zurich and Columbia University. He started as an assistant professor at Maryland in 2017 and was quickly promoted to associate professor with tenure in 2022 and full professor in 2023. He's also a member of the Scientific Advisory Board of Schrödinger and associate editor for the Journal of Chemical Theory and Computation. He's also been recognized for a ton of awards for both research and teaching, and we're very, very excited for Pratyush to be here today. So I'll pass the mic over to Pratyush.
Okay, so can people hear me? Can people still hear me in the back? Because I'm going to jump around, okay?
We'll have to use the mic to get the recording.
Oh, okay, we have to record. I'm going to stay here, so no jumping. Okay, great. So I'm very, very happy to be here. Many thanks to Johnny and to Prudential and to Valence for giving me the chance to be here. This is my first time in Canada, so I'm super excited, even though I'm not spending that much of time, but it's still very nice to be here. And my lab is located in College Park, Maryland, and College Park, just to give you a sense, it's in Maryland, but it's one of the southernmost places, so it's really a suburb of Washington, D.C. On a long run, I can even make it to the White House and run back to my house, so it's that close to Washington, D.C. And I'm a theoretical chemist by training. My group uses a lot of machine learning, but you could perhaps qualify me as a machine learning skeptic. And I was very happy to hear some of the questions that came earlier in the talk on diff-doc and the fitting potentials. And I think I would address some of the questions that even came earlier. And I will try to stop around 45, 50 minutes and hope to have lots of questions, at least leave 15 minutes for questions. Okay, so let's get started. I mean, this title, MD is Dead, comes from a recent American Chemical Society conference that I attended, where someone who I will not name came to me and said, dude, why are you still doing MD? It's a dead field. And I was like, don't tell me that. It's a lot to do there. And I will try to make a case that MD is not dead. It's still very relevant, and there's a lot of scope for creativity. So, and this is my group. We have a very interdisciplinary team of students, people from biophysics. A lot of the work I'm going to show today is done by Dedi Wang, who is joining Genentech, and Connor Zhao, who is going to James Fraser's lab. And then we even have students from physics, chemical physics, chemistry, even an applied mathematician. And I like to claim that my group is the first group in the world to have a postdoc. And so that's Pakora. And and these are the sources of financial support for my group, and that's the conflict of interest. So let's get started. So I'm a theoretical chemist. I like to think about ideas from theoretical chemistry. And I would like to say that this over here is my second favorite equation, or what I think is the second most important equation in all of thermodynamics and StatMech. If there is time later, you can ask me what do I think is my first most important equation. It's not S is equal to K log W, just to let you know, okay? It's something else. So this is the partition function. This tells you that if you can calculate the energy eigenvalues from quantum mechanics, you can basically write down this quantity. It includes temperature through the inverse temperature beta, and you can calculate everything, not dynamics, but everything. You can calculate structure, you can calculate thermodynamics, you can calculate conformations. So, and this equation is also very, very hard to deal with. So if the world was ideal and we had infinite compute time, we could spend all our time computing this equation exactly. And you can already, even if you don't know any thermo or StatMech, you can appreciate why this equation is so hard to calculate, because you have e to the power minus beta e, right? So there will be, it's an exponential. Anytime you have exponential, small numbers and large numbers will come and start to make things complicated, right? You cannot just say, I'm going to stop at this energy value, and you have to tackle everything correctly. So in quantum mechanics, this means you have to get the excited states correctly, and that's challenging. So, and what really inspired my group when I started a few years ago, that a lot of statistical mechanics, which is a field that has existed for around 120 years since Boltzmann, and a lot of machine learning, which I don't know how long it has existed. We don't, maybe we can discuss later, right? Both of these fields, one way or the other, have dealt with this quantity. In machine learning, you might call it the log likelihood function or something else. In statistical mechanics, we call it the partition function, but a lot of the progress in StatMech and a lot of the progress in machine learning has been about how to calculate this partition function, or more importantly, how to avoid calculating this partition function. So one of the most popular free energy methods, free energy perturbation, for example, tells you you can get away by calculating ratios of partition functions, things like that. So just to give you a flavor like of the slides that I'm going to show you next, is that we are really going to think about the confluence of StatMech and AI. And so what do I want to do with this confluence? I want to tackle problems. My group is very much interested in small molecule-based drug discovery. That's a kinase. We are not just interested in understanding the docking pose of a drug. We are interested in understanding how much time does a drug take to dissociate from a protein. What are the different conformations a protein can take? Of late, I have become absolutely obsessed with RNA because when I grew up, I trained as an engineer when I was very young, and I knew that DNA can take a double helix. I never knew that biology can have structures which exist as triple helix. And once you start thinking about RNA tertiary structure, all sorts of crazy structures exist. And it's a fascinating area with lots of scope for creative research that exists out there. And also an area which is fundamentally challenging for brute force machine learning. And then a third part of my group is interested in some more material sciencey problems, such as crystal nucleation, which I won't talk about today. So in all of this, I am interested in predicting structure and predicting dynamics. So if you really think about biology, there are it's like a 4-story building. The first floor is sequence, and there are many people who will tell you sequence is everything. You don't need anything. And then some people will tell you, no, there is a second floor, which is a structure. Sequence might not tell you everything about the structure. Small changes in the sequence space can lead to enormous changes in structure. Then there is the third floor, which is dynamics, that says even structure is not sufficient. You have to understand, how does structure change? When does structure change? And then the fourth floor is environment, right? We can talk anything about a protein and a drug molecule docking, but if you have not accounted for things like pH, temperature, what use is that, right? So I'm not going to tackle with environment. That's really, really hard. I'm interested in the first three, particularly structure and dynamics. And the fact remains that we would like to claim that now AI can be routinely used to solve these things, right? It's less than 4 years ago that AlphaFold 2 came out, and that was a big, big breakthrough. I think I remember reading in Forbes magazine one day. I don't have money. I don't read Forbes magazine. It just showed up on my phone. Like, AlphaFold is the greatest achievement in AI ever. This was the title. That's like a bold title, and perhaps it is true. So this was breakthrough, right? And very soon after that, there was the cover of Science magazine for RNA through method. And this was a method for ranking RNA structures. It was not really for predicting them. This was from Riju Das and others. So, and then in the last 6 months, there has been these were all structure prediction or structure ranking. In the last 6 months, there has been an explosion of methods. And I'm showing you a few of them. The bottom one is AlphaFlow, which said, well, we can train diffusion models on short MD trajectories and start replacing MD. Then there are 2 other methods which are relevant. This one is distributed graph armor out of Microsoft. And this one is neural plexer out of Iambic Therapeutics. And then, of course, up here is AlphaFold 3. I also found it quite interesting that the New York Times article which talked about it had the section over here, chatbots and disinformation, right? So all of these are starting to move beyond, number one, just single proteins, and move beyond just single conformations. They are starting to, AlphaFold 3 makes it very clear that in their paper that they are not able to sample other conformations. And you would think it's a diffusion, so maybe you can get other conformations, but it does not work. These other 3 methods do claim that they can get other conformations. So, and they are exciting methods. They are making progress. And I have my own biases, of course. So I'm not going to criticize these methods, but all I'm trying to say here is there is an explosion of effort. And the other thing to take note is, at least out of these 4 methods, 3 of them have solid industry involvement, right? There is Google DeepMind, there is Microsoft, and there is Iambic Therapeutics. So perhaps the landscape is changing, which makes you worry as a young professor, like how can you even survive in this field when you're going to compete with these things, right? So, but there is scope for creativity. Why? Because there are certain problems which are super important and which are hard to solve even with such state-of-the-art tools. And molecular dynamics is not dead. It's anything but dead. So what do I mean by these hard problems? So I have to give you a big picture, basic biophysics, basic stat mech perspective of proteins, RNA, and crystals. So biological systems, it's not just about one structure, right? So with AlphaFold 2 or with any method, you take a sequence and you predict one structure that exists down in the bottom over here. But we know that biological systems move on some sort of an energy landscape, right? That is the partition function that I showed you over here, the energy eigenvalues that you saw over there. So for a protein, at least for ordered proteins, it's well accepted that there is a funnel-shaped landscape. The most native structure might be very stable. Then there are other conformations which can exist. For RNA, the landscape tends to be glacier. The most native structure is actually not all that different from other competing structures, and just so many of them. Even for the simplest HIV TAR, there are many, many competing structures in NMR database. And crystal nucleation is a fascinating problem because it really combines the worst of protein and RNA. When a crystal forms, you have a sharp minima like a protein, and when you form a glass, it can look like an RNA. So these are three energy landscapes, and if we really want to say that we want to do biophysics or drug discovery, which can help us sample these non-native conformations, and why do we care about non-native conformations so much? So if you're in the audience here, and if you have read this book, by Tolstoy called Anna Karenina. Can you raise your hand? Okay, good, many people. And who here would claim that they remember the first sentence of this book? I buy you a beer. Go. Bravo, you get a beer. So I have the protein. So I have the protein kinase version of Anna Karenina, which is called Anna Kinesina, and that says that all active kinases are similar, all inactive kinases are inactive in their own way, right? So if you can tackle these non-native conformations, maybe you can start exploiting attributes which make your drugs specific and things like that, right? And these are underrepresented in the protein, in the PDB. So, and these things matter, right? This is a review by Ruth Nusinov at NCI where she shows that subtle changes in active versus inactive, not just which is the most stable state, but how much is the difference in the free energy can lead to phenotypes where you have cancer or neurodegenerative diseases, right? So free energy is relevant. It's not just about one structure. It's really about understanding the ensemble of structures and the relative weights. So how does AlphaFold do on kinases? Terrible. This is AlphaFold 2. Most of the results would apply for AlphaFold 3 also, but for obvious reasons, I cannot even check that. So this is AlphaFold 2, where you take a kinase, and for those of you who don't know a kinase, it's the second most important drug target after GPCRs. And it's 5% of the genome, and much of the features in kinases are preserved. So you have an alpha C helix, you have this activation loop, the disordered part over here, and you have this DFG motif. So for a kinase to function, so what kinase does is it takes ATP and it changes it into ADP, right? So the ATP molecule has to come and bind right in the middle over here. For this to happen, everything has to be positioned right exactly, okay? So it's like the happy family being happy in exactly the same way. But every once in a while, this activation loop can move by 12 angstroms. It can flip all the way over here. The DFG motif can flip. The alpha C helix can break some salt bridges and go to the side. And a lot of blockbuster kinase drugs, actually, including possibly the most famous kinase drug, Gleevec, does not bind to kinase in the active conformation. It binds to it in the non-active conformation, the inactive conformation. So different kinases, such as DDR1, ABL, and SARC, can show different probabilities for being in the active conformation or being in the non-active conformation. So you can take AlphaFold 2, you can do what is now known as the MSA subsampling method, which literally means you weaken AlphaFold. You tell it, don't look at long sequences, look at short sequences. And by doing this, you can generate lots and lots of structures, thousands of structures if you wish. And since if you subsample the MSA, there is diversity. You naturally get diversity in this. You can do it with other methods like AlphaFold AF cluster also from Dorothy Kern and collaborators. And turns out all kinases kind of look the same. Even when you get diversity, it's not able to tell you that kinase 1, you can say that, oh, maybe the green line is smaller in SARC than in ABLE than in DDR1, but not really. Statistically, there is no significant difference. Is this representative of reality? Not at all. For DDR1 kinase, it is well known that this pie chart should actually look equal. There should be, all three conformations should be equally possible. And AlphaFold simply cannot do that for you. For RNA, I'm not going to speak much for a variety of reasons, but I just want to point out this slide that CASP competition, which AlphaFold won a couple of years ago, CASP 15, of course, a big part of the reason was the number of structures that exist out there in the PDB, right? For proteins, I need to update this slide. It's more than 200,000 structures now. For RNA, even though the number of RNA is much more than the number of human proteins, the number of structures is less than 2,000. So in CASP 15, the assessment published by the organizers was that prediction from deep learning approaches did worse than non-deep learning approaches, like across the board. And AlphaFold 3 also works on RNA. You can just go and check yourself. And they also published most of the structures. It does not do well. In CASP 16, it's underway. We will see. Maybe deep learning will change everything. I guess we will know by November. My group is also taking part in it, so I'm not trying to speak too much about it. And finally, if I have some time left today, the third challenge I would like to show, and this came up in a question answer session in the last talk, can you just take short molecular dynamics trajectories and train machine learning models on it and get something done? No, it's not going to work. This is a synthetic peptide made by Peter Ham in Zurich. It exists as a left-handed helix, as a right-handed helix, okay? And it's symmetric. It's engineered to be symmetric. So if you take a short MD trajectory where there is asymmetry, the left-handed is preferred slightly over the right-handed, and you train, and we did this work when there was no ChatGPT. We did it with LSTMs, and the student moved on. He joined Sharon Glotzer's group. But I'm trying to revive this project now because there is so much interest in this. So the idea being that, and you can do this with transformers also, any language model, if you train it on a trajectory where there is some error, the language model is going to commit that error to memory. There is no way that it will know that this is a systematic error. So if you take this trajectory and you train an LSTM and generate a longer trajectory, you reproduce the same error. So shorter trajectories are not sufficient. And if I have time, I will show you how using, again, statistical mechanics, we can solve this problem. But mostly today, I want to focus on proteins and tell you how we can speed up molecular dynamics. There are many people working in this area. I will show you some of the ideas that my group has been working on. So my group develops lots and lots of methods. All of them are online on GitHub. They are not easy to use. So I might be the only person in today's session who will tell you, nope, my methods are not easy to use. You will have to suffer through it. It's complicated. There are demos. You will have to learn it. And perhaps deliberately so, we don't want it to be super easy to use because that's when you make mistakes. So there is a learning curve. These are not just, you put the, that said, it's amazing how much easier to use they have become with time. It's so, but yeah, that warning, you should check them out. And so we have three classes of methods in our group. One is this information bottleneck approach, which I will mostly talk about today for speeding up sampling of proteins. Then the second one uses diffusion models. Notice our first paper on diffusion model came out in 2022. We submitted it in 2021, which was well before DALI and stable diffusion and things like that. So, and then, so it was cool. And that's what we use for RNA. I won't be talking about that today. And if I have time, I will talk about this third method. So let's get started. So what is molecular dynamics? It's simply solving Newton's law of motion, right? If I could model, and Gianni showed it very nicely in his talk, you have the force fields, right? So you just do force is equal to mass time acceleration. If you have 100 atoms, the number of F is equal to ma equations you need to solve will become 100 multiplied by 100, right? 10 to the power 4. Approximately. Most of the times you don't have, it does not scale as n square, it scales as order n log n, because electrostatics can be done in a Fourier space. So there are tricks, people play with it. But the fact remains that it's a differential equation. Newton's law of motion is a differential equation that you need to solve. The time step is femtosecond. And you cannot play tricks with time. You can play tricks with larger systems where time is, it's ruthless. You cannot play tricks with time. So people have tried to change the acceleration, the integration time step in molecular dynamics, but then the MD becomes completely unreliable. So this number of 2 femtosecond is, some people say even that is too slow or too much. You should go down to 1 femtosecond. And if you're doing quantum molecular dynamics, the time step becomes 0.1 femtosecond. So imagine in a protein, in a box of water, you have 100,000 atoms and you're solving 10 to the power 8 equations of motion to advance your clock by 1 femtosecond. That's kind of expensive, right? If you want to study things like protein conformational change or crystal nucleation or drug binding, they happen really, really slow. So with high performance computing, with GPUs, I guess my students told me I should change Call of Duty. No one plays it anymore. It's some newer games, but whatever. And with very specialized computers such as Anton, which is David Shaw is a billionaire who put in much of his money into making one computer which does molecular dynamics very, 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 very fast. And it's been phenomenal because they can reach up to millisecond in a few weeks. But the problem even with that is you get one event, right? You have a protein folding problem and you get one event. That does not tell you anything about the statistics of what's going to happen, right? You observed it once. Does that tell you everything about what might happen? No. In order to get the statistics, you have to really simulate the slowest processes also and get that right too. And this is really hard. Why is it hard from a more fundamental perspective? So here I'm showing you a typical molecular dynamics trajectory projected along some coordinate. So what do I mean by that? So for a drug unbinding problem, the coordinate could be the distance between the drug and the protein, right? So this is the bound state, this is the unbound state. Of course, you would not use a drug where the free energy looks like this because this would mean the delta G is 0, right? KD is not there. So for a protein conformational change, the projection could be the number of native contacts in the protein, right? The radius of gyration, something like that. So what happens in molecular dynamics is you are trapped in one basin. You never get to the other basin because there's a very, very high barrier that you need to surpass. Now, there are these enhanced molecular dynamics methods which allow you to deal with this problem. How do they do that? One of the ways of doing it is to fill up the energy landscape. So wherever you are going, you just flood it with sand, or computational sand, whatever you want to think of it. Technically, it means you add a small repulsive potential, so repulsive bias in your force field. You keep building it as a function of time, and effectively the height of the barrier starts to feel lower. And once you go to the other side, again, you start filling up this one, the height of the barrier becomes smaller, and you do back and forth. Notice I have changed the coordinate here. On the top, I said some projection. On the bottom, I said reaction coordinate. What do I mean by that? Some projection means literally any projection. If I'm trying to describe a position on the Earth, the latitude and longitude are good projections. They tell me where you are located, but they are missing out some information, right? They are missing out the elevation, for example, how high above sea level you are, right? So projections start to miss out information. A reaction coordinate, on the other hand, is a very special type of projection. Information about all slow things that are relevant. Okay, so it's a term that most of us have probably seen in our high school chemistry textbooks, right? You have a reaction, and reaction coordinate tells you how much have you progressed along the reaction, right? And we rarely think about what is a good reaction coordinate. For drug unbinding, okay, is the distance between protein and the drug a good reaction coordinate? Maybe. How about the water molecules? We know a lot of good drugs have to displace the water molecules from inside the binding pocket. How about protein conformational change? So it becomes complicated. And these enhanced molecular dynamics methods, one way or the other, require you to know a priori what is the reaction coordinate. So that becomes a problem. You're trying to solve a problem where you don't know the mechanism and it's too slow to study. And in order to speed it up, you need to know the mechanism. How do you do that, right? So the movie I showed you here is a method known as metadynamics, which was invented by my postdoc advisor, Michele Parinello. There are many, many other methods. Okay, second beer opportunity. Who here thinks they know the full form of all these enhanced sampling methods? Maybe. I could write a couple more and then you would be like, I don't know. So it's a zoo of enhanced sampling methods. So Johnny mentioned I'm editor on this journal, JCTC. I handle something like 300 papers a year, and every week there is 2 papers with a new enhanced sampling method that says, we don't need reaction coordinate. We can speed up the simulation without knowing the reaction coordinate. As a critical community of people start using the method, turns out, no, you need to know the reaction coordinate. There is no shortcut. And we can argue over whether these other methods need it or not, but all of them, one way or the other, know it. So this becomes a very hard problem to how to figure out this very special projection without solving the problem. So, and let's get a bit more technical and give you intuition. This is a school, right? So it's not just about me telling you what I have done, but trying to help you get a basic sense of why the things are challenging. So I will start with a picture of a free energy along some coordinate. And I am marking 6 states over here, okay? And my question for you is, and this will involve everyone raising hands, okay? So be ready. My question for you is, does this point over here, do you think this is a transition state or not? If you think this is not a transition state, raise your hand. I see few hands. You have to raise your hand one way or the other, okay? So there is no escaping. Okay, and if you think this is a transition state, then raise your hand. So we would agree that majority people went for this is a transition state. So I have a conjecture here. Every time I ask this question, more people raise their hand to the second answer. This is a conjecture. People wait to see what's going on. Turns out this is not a transition state, okay? So the first group of people were correct. Why this is not a transition state? Because this is a projection. You have removed other degrees of freedom and you don't know what are these other degrees of freedom. In this case, this was a projection from these 6 states where I literally marginalized out R2, okay? So I had P of R1, R2, which is the probability, and I'm going to do integral of P of R1, R2, dR2, okay? I'm just going to remove R2. And it looks something like this. So now what you think is the barrier between 4 and 2 is misleading, right? There is no direct pathway between 4 and 2. To go from 4 to 2, you have to go through 5, which is completely misrepresented here, right? So this is what I mean to say. R1 is a projection. There is nothing wrong with the thermodynamics in this picture. R1 is an okay projection. R1 is not a good transition. It's not a good reaction coordinate. And the transition states that you see along R1 are not good transition states, okay? So this might make you feel hopeless. Like even for such a simple problem, if I didn't know the missing degrees of freedom, it can be so wrong, right? So after this talk, hopefully anytime a colleague of you comes and tells you this is the barrier, this is the transition state, you will be very, very suspicious, okay? You'll be like, what did you remove? What about the other degrees of freedom, right? Always ask about that. So, and now we have this zoo of machine learning methods like TSNE and UMAP. Here I'm showing you projection using TSNE and UMAP of a higher dimensional object. Can anyone guess what is this higher dimensional object? It's a mammoth. And it projects it into something, right? You have no clue that it's a mammoth. It's something else, right? So there is all these other methods which do projections and which make nice clusters. And it's natural to think that just because you have clustered states away from each other, they carry some mechanistic information. And there is a whole lot of debate that happens in the bioinformatics community, for example, about TSNE and methods like that, right? You have made these clusters separated from each other. Does this really mean that data points in the cluster are far from each other or not? We don't know. So we have to be careful. Just because we have this clustering method does not mean we can use that clustering scheme to speed up my dynamics in metadynamics or a method like that. So we have to think a little bit about mechanism. So how do we do that? So for this, my group built up on work done by some very smart people, specifically Shannon, the most famous Shannon of information theory in 1940s and 50s. He really invented the field through something known as rate distortion theory. But more recently, Bill Bialek, who is a biophysicist at Princeton, around 30 years ago, kind of revamped Shannon's idea in the terms of something known as an information bottleneck. And that's what we are going to use today. And I will show you how such an information bottleneck can be learned through deep neural networks and how it is a clustering scheme that actually captures something about the reaction coordinate, which can then be used to speed up molecular dynamics. So what do I mean by an information bottleneck? So what I mean here is I'm trying to come up with a model of a system, okay, of some complicated system. So let's take an example. Let's say I know the temperature of all cities in North America today, okay, and I want to predict the temperature in Montreal tomorrow. So it's a very high dimensional input, and the output is just one real valued number, right? What is the temperature in Montreal tomorrow? Or maybe the input is a bunch of photos, and the output is whether the photo contains a dog or a cat or not, right? So just binary. So it could be anything, any model that you can think of. Bialek says that a good model should have 2 properties. Number one, it should be very predictive as possible, right? If your model is not doing a good job of predicting what you wanted it to predict, then what is the point of that model, right? It's garbage. At the same time, the model should also be compressive. That's a bit more nuanced to understand. Why do you want the model to be compressive? The best way to understand is to think that, let's say, is in North America today, and you build a model where on this information, you can predict the temperature tomorrow in Montreal. Now, let's say I add 1000 degrees of freedom to this model, which are just white noise. There is no information in that, right? Your model should be able to say that this noise degrees of freedom don't carry anything. I don't need to consider that, right? It should be able to predict the signal from the noise. So you get a double funnel structure where you're compressing the input as much as possible while still being able to predict the output as well as possible. This can be written down mathematically as a difference of two mutual informations. You have mutual information between the input and a bottleneck that you want to minimize, and you want to maximize the mutual information between the bottleneck and the output. And this idea really comes from a wonderful paper by Alex Elemy at Google, known as deep variational information bottleneck. That's what we are using over here. These are generalization of possibly one of the most famous deep learning architecture, variational autoencoders, right? In an autoencoder, the input is the same as the output, right? And this beta term that you have over here is 1. Beta VAE introduces this beta term, and that leads to disentangled representations and things like that. So this is a generalization of a beta VAE. That's one thing you can think of. So what we showed specifically in this paper done with my student Dedi Wang is that for molecular dynamics, if the input is the high dimensional input trajectory and the output is the future state of the system, and we will take some time to think about what do I mean by state of the system, then the bottleneck approximates the true reaction coordinate from chemistry. What do I mean by true reaction coordinate? So there was this very famous chemist or physicist, Lars Onsager, who won the Nobel Prize back in the day for work he did during his PhD. And Onsager in 1930s introduced this notion of something known as a committer. A committer can be shown to be the perfect reaction coordinate in chemistry. Okay, and I'm not going to talk about what a committer is. You can go and look it up, but we know in chemistry a theoretical way to calculate the reaction coordinate. It's just that it's very hard to do it. What we show in this paper down here, that this approximates the committer. And okay, so now let's get more technical. So another difference we have from variational autoencoder is biological systems are not made of only one state, right? A protein conformational change problem has many different conformations that exist. A drug unbinding problem has at least 2 states, the bound state and the unbound state, and possibly different intermediates that lie around the path. So in a typical variational autoencoder, you use a single Gaussian prior for the latent variable, right? Which is what makes them so easy to train and easy to use. Here we use what is known as a VAMP prior, not to be confused with Frank Noyes VAMPnet. This VAMP prior also comes from Waxwelling, where he says it's basically like a mixture of Gaussian. So for a biological system, you do not know how many states are there. So you assume a mixture of Gaussians for the prior. You say, well, I don't know how many states my biological system has. Let's say it has 1000 states. And then you can iterate through the process. You can start with an input. You can try to predict the future state of the system, and you will find that most of the states don't have any population. They disappear. And the method automatically learns how many states your system has. Another important and technical thing here is the inputs we take are not all atom coordinates of molecular dynamics, because then we have to worry about equivariance and things like that. We take features such as distances between atoms or dihedrals, which do equivariance in a cheap way. Okay, and we can get away with that. Of course, there is no free lunch. We lose out on all atom information. We do what is known as poor man's back mapping to recover that, and I can talk about that later. So this is the framework. I have a high dimensional input molecular dynamics trajectory. I can learn such an information bottleneck experienced as a mixture of Gaussians, which allows me to predict the future state of the system. And this is the reaction coordinate, which won't be misleading you like that picture, which misled all of you, right, about the states. This will actually be correct. So let's look at a very simple system to develop some intuition. So this is a 4-state system. There is nothing missing here. Okay, these are the only 2 coordinates, and you have, this is the free energy, which means that deeper the blue color, higher is the probability of you staying over there. Red means don't go there, very low probability. Okay, very high energy, very low probability. So what we do, we don't know. So let's say we have not sampled anything from system. This is the ground truth, right? So let's say we just mask that part. And all you have done is to generate some data from it. You assume there are 10 states. In this case, there are not 10. If you first look at it, it looks like there are 4 states. But that depends on your timescale resolution, right? Because there is a very high barrier over here. There is a second higher barrier over here, and there is a lower barrier over here. So if your time step is super long, then this is not a 4-state system. This is probably a 2-state system, because this barrier around x is equal to minus 0.5 and the barrier around x is equal to 0.5 will not be relevant. You will be equilibrating between these 2 states. So we start with the 10-state definition. We pick how far into the future we want to predict. This is a parameter that you can take. This is different from those of you who know Markov state models. In Markov state model, you take your time lag as long as possible so the model becomes Markovian. In our case, every time lag gives you new physics about the system. So we pick some time lag and we learn how many of the states actually have probability left in them. And we find that for this time lag, 6 of the states disappear and you recover 4 states. And the key idea here is that the time lag is working as a filter for metastability. The idea being that now, if you increase the time lag, if you're trying to predict more into the future, the system will be equilibrating between states 1 and 3 also, right? This all sounds very abstract, but I'm saying something very simple here. In the long run, we are all dead, right? In the long run, all of us have only one state, death. That's the final solution. But if you're trying to predict for what's going to happen 1 hour into the future, then there is many states that a system can take, right? So any given biophysical system, the number of states that you have in that system decrease as a function of your time resolution. If your time resolution is 1 picosecond, if your time resolution is 1 femtosecond, then you need to account for every water molecule, what is the state it is taking, right? But if your time resolution is 1 microsecond, then the water molecule motions average out, right? That's the whole idea over here. So for enhanced sampling, we don't know a priori which time step to take. There is a bit of art that goes into there. And in fact, every time step carries its own information. It's really about which barrier are you trying to enhance the movement over, right? That's what it comes to. And yeah, and so like I said, if you take smaller time step, you get 4 states. If you take a bigger time step, you get 3 states. If you make it even bigger, you get 10 states. You can go to our GitHub and play with this. You can run it in Colab. There is literally nothing to do. You don't even need Colab Pro. It will run right away. It does the MD and everything. And you will see that if you make the time step even longer, you are left with only 1 state. That's it. Everything equilibrates, right? So, and yeah, like I mentioned, it approximates the true reaction coordinate. And yeah, so with that, now I can show you the framework for enhanced sampling that we call RAVE. It was one of those methods where it was a desperate attempt where the acronym came first and the full form came later. We're like, we are going to call it RAVE for a variety of reasons. And then we tried very, very hard to come up with a full form, as you can see. So RAVE says that you start, the idea in RAVE is you start your molecular dynamics. It's a short trajectory, or it's a trajectory biased along some trial reaction coordinate, which might not be a good coordinate, but you do something, right? You generate data one way or the other. You take this, you learn an information bottleneck. Notice I'm showing a linear encoder. So by the way, I guess I forgot to mention one very important thing. You learn a map here from the input to the bottleneck, and you learn a map from the bottleneck to the output. Just an encoder and decoder. Both can be deep neural nets. And here, until the data quality has improved, we stay with a linear encoder. Number one, it's harder to overfit a linear encoder as relative to a deep neural network. Number two, it's easier to interpret, right? You can go and see if something does not make sense. Number three, linear models extrapolate better than deep neural networks, right? So if you have a linear model, what you're trying to do is to take this and increase the sampling, right? So neural networks are fantastic, and this is a problem which is happening in neural network potentials, for example. They do wonderfully well if you keep using them in the regime where they were fit. If you take it to another temperature, everything goes away, right? And that's a problem. So when we first started doing it, we thought, oh, we will quickly switch away from a linear encoder. And now we have come to appreciate linear encoders more and more. The decoder, you can make it as deep as possible. There is no problem. You can make it, generally find 2 or 3 layers is sufficient for this. And the inputs are features. They are not all atom positions. We have been able to change it through a graph model where we can get away with that. But for now, we just take features which depend on the system. So now we have this information bottleneck, which we know approximates the committer, the true reaction coordinate. We can put it in some enhanced sampling method, metadynamics, umbrella sampling, weighted ensemble, whatever you want to do, and get a new trajectory. In this case, this is a ribo switch. And we are looking at how does a small molecule dissociate from a ribo switch, and we keep iterating. When do we stop the iteration? Well, the naive way to think about this would be to say that when chi, the bottleneck, stops changing, right? But that is hard to quantify. If you have 2 degrees of freedom, you could just be rotating in this plane, right? You could have some variance and you won't be able to say whether it has converged or not. So what we do is to think about this again from a thermodynamics perspective, especially in this method metadynamics. When you're biasing a system, taking it from point A to point B, you are doing work on the system. And thermodynamics tells you, second law of thermodynamics, that the work done on a system will always be more than the free energy difference. You can never go lower than the free energy difference. So as we improve the reaction coordinate, we find that the work done on the system calculated through metadynamics decreases through every round. And at some point it stops changing, it converges. And that's when we say, okay, we cannot do any better with the feature set that we input over here. Maybe if we had better features, we could have done better. And that's when we publish or draw information from whatever we have done. We test our methods on lots of model systems. Often in molecular simulation, a lot of people do this thing, which I find very strange. They work on really small systems, alanine dipeptide, and they immediately jump to coronavirus. And that's dangerous, right? The really small systems, we already know everything that happens, right? We already have intuition. In coronavirus, we know nothing. So those insights can be useful, but as a method developer, it's very important to have these hierarchy of systems which are increasingly complicated, right? So these are systems where the dynamics happens on the microsecond to tens of microsecond time scale. So they are slow enough that you can call them a rare event, but they are fast enough that you can generate long benchmarks, again, using the same force field, right? Anytime you're doing molecular dynamics, the force field will have enormous effects on what you're doing. So comparison to experiment might be a lucky thing that is happening, right? So you have to compare apples to apples. In the end, we of course want to compare with experiments and help experiments, but as a method developer, these systems are very useful. And I said I won't talk about crystal nucleation. I just wanted to show the slide because in drug discovery, for example, there's a lot of interest in predicting the right polymorphs. So in this paper, in the bottom and these others, we showed how RAVE can be used for completely different problems in material science and really getting crystals and different polymorphs to form back and forth and things like that. So, but now in the remaining 5 minutes, then I'm going to shut up. So I won't have time to talk about the long short-term memory because I want to leave at least 10 minutes for question and answer. Let's talk about AlphaFold. So I mentioned earlier that with subsampled AlphaFold, and this was really done by Diego del Alamo and Hassan Mekharab and collaborators in this eLife paper. They were the first people to publish this, that by reducing the MSA depth, you can start generating new conformations, right? So if you have a very long MSA depth, you get few conformations, and you have to run AlphaFold thousands of times to do this, which you cannot do on a web server on AlphaFold 3, for example, right? You really need to be able to run it lots and lots of times. You're using it as a hypothesis generator, right? So these days, when I got tenure, I was like, okay, what do I do with my life? I don't want to just sit writing more grant proposals and fighting with other people. I want to do more coding myself. And that ChatGPT came. It was wonderful, right? Because Stack Overflow is good, but it's still, if you have really left coding for a long time, ChatGPT and Copilot make your life much easier. But you have to take it with a grain of salt, right? You're asking it to do something. It's a hypothesis. It's one way to do something, right? And maybe you like one of the ideas, maybe you don't like them. So that's how we are treating AlphaFold. In this work, what they showed is by reducing the MSA depth, you can generate lots and lots of structures. That's good. You are getting conformational diversity, but that's not quite there yet, right? All you have done here is to generate too many conformations. What are you going to do now? Are you going to dock your ligand against every single of these conformations? Are you going to do your FEP calculations on every single conformation? That's the compute time will blow up, right? So Brian Schoikert at UCSF has been doing this incredible work with ultra-large library docking, right? Billions of compounds, and he does it on one conformation. If Brian really wants to sample different conformations, he needs to know which of these conformations should I try, right? He cannot do it on 100 conformations. That's not going to work. So these are unranked hypotheses, and we need to have a way of ranking them. And that's where we have AlphaFold to RAVE. AlphaFold to RAVE was basically done by my PhD student Akash and Bodhi Vani, who is now a machine learning scientist at Prescient Design. And now my student Zinyu Gu, my postdoc Zinyu, is leading this work. So the idea is you have a sequence, you generate lots and lots of structures using reduced MSA AlphaFold. You can even use AF cluster for this. And you shoot short MD trajectories from this, as short as you can. All you do is to remove some of the errors that might be there. You get the waters to settle a little bit. So these are discontinuous trajectories, right? You have never seen a movement from point 1 to point 2. Now you learn an information bottleneck from this. It's not going to be very good, but it will have some information. You do your bias sampling, you generate new conformations. At this point, you can take these new conformations because there is some chance that they are thermodynamically meaningful. You can even use them as a template to pass it back through AlphaFold, or you might not iterate at this step. If you can see my cursor moving, you might just iterate between this step and the rest, right? So you keep iterating. You use a weakened AlphaFold or some other method. It could be a diffusion method also, for example, if you want, to generate many hypotheses, and you use enhanced molecular dynamics to rank them. You cannot rank them with just short molecular dynamics, because over here, all you have done is to just look at how things jiggle and wiggle in their local energy basin. You don't have any sense that this structure over here is so much higher in energy than the structure on the bottom. For that, you need to do enhanced molecular dynamics and get a sense, because the barrier is very, very high. And with this, for example, for DDR1 kinase, we can get this DFG loop to flip. We can compare our benchmark with John Cordera's work on folding at home, where he did ultra long MD, and we used the same force field. And we see that with AlphaFold, you see, first of all, that DDR1, AlphaFold would just tell you that in is more stable, which is not true. Secondly, AlphaFold does not get the prediction of mutants. With AlphaFold RAVE, we can reproduce with much less compute time what John found through Markov state models. And my final slide on this is regarding docking. So it's cool that we can dock ligands against structures generated from AlphaFold. But you really need to ask, did you need AlphaFold to generate that structure? Or could you have just picked up that structure from somewhere else? And often, in a lot of the papers that have come out in the recent years, it's not clear. Recent years, I mean, the last 3 years, like, did you really need AlphaFold to run that docking campaign, or did that thing already exist? For example, for a kinase, we already know it exists as a DFG in. So in order to predict a DFG in structure for a kinase, you don't need AlphaFold 2. Some older homology model would have done the job for you, right? And specifically for DFG out, and this paper is in eLife, it's out for review, and it's up on arXiv also. We show that if you're trying to, there's a lot of information here. It's a very, very new thing. So I just took a screenshot from the paper. But the point here is that, notice here, this is the activation loop, right? So the in kinase, in form, the red form is where the activation loop sticks. For the inactive kinase, the activation loops moves by 12 angstrom. It's a huge, huge move. And NMR doesn't get this right because it's disordered, right? So it's not there. It's not in most of the PDB structures. This loop is, you can go and look at it. This is where information is missing. So you cannot train on this. And on the top over here, you can see the DFG motif. So the thing has to move, and this DFG motif then has to flip. So what we find is that for ABLE1, ABLE kinase, if you run 1000 AlphaFold 2 or AF cluster, some of them, 10 of them, have the activation loop actually moved the right way. Now, if you focus on them, none of them have DFG flipped. So if you were trying to bind, this is a purely retrospective study, okay? There is nothing prospective here. What we're trying to do is to take drugs like imatinib and ponatinib, which are type 2 binders. They bind to inactive kinases, and see what would it take to get structures from AlphaFold 2 where we can bind them. For ABLE kinase, you would never get a structure where you can bind them. For DDR1, you can get some structures where you can bind them, but again, you don't know which of these structures should you actually bind to, right? There are 1000 structures. You're not going to run your docking on all 1000 of them. So with our RAVE method, we can get a free energy landscape, and we can just pick the most stable structure in the free energy, and we can dock to it, and we get ligand RMSD, which is well within 2 angstrom. While if you were not doing RAVE and you were trying to bind these type 2 structures, the RMSD never goes under 8 angstrom or 9 angstrom. And now we are trying to generalize this approach to not just kinases, but GPCRs and other things. And I'm happy to collaborate with people on this if they're interested. And I'm not going to talk about the LSTM work. these slides are online, so you can just, I also put it on Twitter. If you're not on Portal, you can just see them there. So let's conclude. My main take home today was molecular dynamics started around 60 years ago. The first molecular dynamics paper was around 1964. Depends on who you ask, from Anisur Rahman, where he simulated 1,000 argon atoms, rare gas, talking to each other. And we have come a long way from them. It's been 60 years. Now we are doing billion atom molecular dynamics. And AI is not going to reproduce molecular dynamics. AI at best in all the AlphaFlow and other methods, all I'm seeing is it's doing jiggles and wiggles. It's taking a short MD and reproducing that. It's simply not clear why should we use these methods over MD. 100 nanosecond MD trajectory is not 10 times better than a 10 nanosecond MD. This is not obvious to a lot of people. If you think about it, it will become obvious, right? Because if all it's doing is to jiggle and wiggle better in that energy basin, right? So you have to see the rare events of interest, and these are emergent phenomenon. If you train a machine learning model on a 10 nanosecond MD trajectory and run it 10 times longer, it's just going to reproduce that thing 10 times more. It's not going to show you behavior that you would have seen in the MD, the behavior that would have emerged if you had run your MD 10 times longer. And new physics emerges as you keep running your MD longer and longer. And you cannot generate such training data. So is the answer just to run lots and lots of MD? We all get together and run more MD simulations unfolding at home or wherever, and then train machine learning models on that? Yes, we can do that, and maybe these models will be useful. I don't know how much useful they will be relative to MD. The amount of data you will need to do this MD would probably make these models useless. This is why enhanced molecular dynamics is going to become more and more relevant, because it allows you to generate data which is quick, cheap, and then the machine learning model can go and supplement it. And today I showed you only the RAVE method. I didn't talk about path sampling, and all my codes are on GitHub, and I'm happy for questions.
All right, if you have a question, raise your hand. Okay, here we go.
Thank you. That was an excellent talk. I was just wondering, I don't know if I missed this somewhere, but how did you guarantee that your model, the RAVE model, is going to go through a physically possible transition state as opposed to some unphysical process where like atoms start clipping through each other or something?
Yeah, so that's another advantage of not using atoms as the input features, but using distances as the input features. So in RAVE, and this is another reason why we are not, so this is the problem which is happening in DivDoc and other things, right? The things become crazy. But if you don't input all atom coordinates and you just input features, this problem goes away. You still might get, so that's one reason. And secondly, we are actually generating thermodynamic weights. So the method tells you that things that are going crazy have a very low weight. And this is how we solve this problem, but we lose out everything. So how, in the end, we want all atom trajectories, right? So to go back from the feature space to the all atom space is a non-trivial problem. And we are doing it in a very cheap way, which is not the best way.
Hi, thank you for the nice talk. When it comes to using machine learning for MD simulation, and it's not guaranteed that the machine model is actually learning the physics. It's very likely that it's just maybe memorizing the training data.
Yeah.
And even though you use a equivalent model, it's still not guaranteeing that for example, the energy is conserved or not.
Yes.
So yeah, my question is how to encode the symmetries or any conservation laws so that the trajectory generated by the machine learning model actually makes sense to the real world, or obey the requirements for some examples of the simulation.
And this is why I'm coming back to this slide. So look at the loss function here. The loss function itself is physics inspired, right? We are not trying to regurgitate all atoms in the system. Here we are using a deep physical concept of metastability. We are saying that biophysical systems in an energy landscape are very boring, right? If you really think about a protein, what does a protein do? Not much for a very long amount of time. All of a sudden, everything gets together and it gets into another shape. So what this, by introducing a time filter over here, we are saying, don't worry about what's happening when you're trapped in one state. If you're trying to do that, you will learn, you're just learning the noise. We don't care about that. So that's one way. And the second, more broader way is dynamics. I think a lot of machine learning models are training purely on structural data. The more we start training on dynamics, the methods will start becoming more and more accurate. I think dynamics is like a regularizer, for lack of a better word, which cleans things up.
Hi, thanks for the talk. When you're using RAVE at the end, you started with different conformations from AlphaFold using the
Yeah.
subsampling.
Yeah.
Have you considered using other techniques to get those initial conformations, like normal modes analysis or something like that?
Yeah, we could definitely use that. So think about, it's amazing that you bring up normal mode analysis, because I have always thought of the end state of a diffusion model as a normal model, right? Everything is Gaussian and not talking to each other. So maybe a diffusion model is taking you to that, right? So these other states could be coming from a normal mode analysis. It could be coming on, some people are doing this to take the structures coming from AlphaFold and fit a diffusion model and then generate lots and lots of structures from the end of the diffusion model. So we can do all sorts of things. There is no problem. AlphaFold 2 was so democratic, right? It was so easy to generate these structures without doing anything. That's why we started using it. AlphaFold 3, we cannot do that. So maybe we will go to normal mode analysis.
Hi, thanks for the talk. I'm wondering, as an ML skeptic, but also MD expert, how often do you find yourself using neural network derived force fields?
As of now, 0. Should I?
I don't know. Why don't you use them?
Because, so which one would I use? I think about water. So my good friend Francesco Pesani has a fantastic model for water known as MBPol, which does water very accurately. But then I think, well, the protein force field have been changed over the decades to account for all the errors in the previous water model. So I have to go and change the protein also, right? So I think there is a barrier. Maybe the time will change, but I'm not convinced yet, to be very honest, that I should go beyond physics-inspired force fields. I'm watching with a sense of curiosity and seeing what's going to change. I'm open to it.
Thank you.
Yeah, we'll take a last question here.
Hi, thank you for the great talk. The slide is perfect. So I was wondering, as a person that doesn't know a lot about MD, how long does it take to actually rank all those AlphaFold structure that you generate with enhanced sampling? Like, how long would it take?
Yeah, so much slower than running AlphaFold. And, we are still scaling up these projects on different systems. So for now, it's okay, so we can answer that question more technically, more rigorously. First problem is feature selection. That is still a bit system specific. For kinases, we know what type of features to do. For GPCRs, we have some sense to do. So that problem we have solved. So if you come with a new kinase or a GPCR, we have some sense of telling you the feature selection part. If you come up with a completely new protein, then I don't know. So that could take weeks in just getting intuition into what are the good features. Because you don't want to go predicting all possible crazy conformations, right? You want to have a sense of where the features are. So that part can change from a day to weeks. Secondly, the MD, like I made the point, running long MD is not really needed here, because in this training set, if you ran a 10 nanosecond MD and instead you run a 100 nanosecond MD, you're not getting all that much information. So there we are very fast. We can just get a very, very quick MD. So overall, I would say this protocol, we can get it done in the order of weeks, but we are constantly working on improving this and making it faster.
All right, thank you.
Might be able to squeeze in 1 or 2 more questions if anyone has a question.
Yeah.
Okay.
I was actually wondering, when you talk about reaction coordinates, I keep thinking on chemical reactions. How's the possibility of applying those, for example, to substitute an IRC calculation and really calculate the transistor state of a chemical reaction, maybe using QMMM or I don't know.
Yeah, we could. I do have, I did work with Parinello. So I have one paper on Parinello microdynamics where I did that. I have one on Born-Oppenheimer, but somehow we did not pursue that. In principle, these methods could work on the Born-Oppenheimer level. If you're going to path integral, then it becomes more complicated, because truly the reaction coordinate for a quantum system is not localized, right? Quantum mechanics is all about delocalization. And the reaction coordinates I'm learning here are combination of local features. So if you want to stop at a Born-Oppenheimer level, it'll work. And I think that's normally what people do. So QMMM, I think it can work. I'm interested in pushing it in that direction, but we haven't done it myself yet. We can check, yeah, and we can talk more about that. So send me an email.
Okay, last question before we break for lunch.
Thank you for the talk. I don't have much experience with MD, and I'm trying to understand if the features would all be global features, or would they also be atom level but still MD?
Yeah, we use all sorts of things. So now we are also starting to do it with graph neural networks, for example, where we have local features which we pool in such a way that it starts telling you something about global. So long story short, it can be both, but sometimes it helps to know which one you want to prefer. So it's not fully automated yet. And that's the advantage of doing it directly from atoms. You won't have to worry about things like this. But in practice, we try to make a sense a priori. So actually, I can show you with this kinase problem. For kinase, the degree of freedom that matters is a big movement of the activation loop, right? The whole loop has to move. So this is a global movement. So in this case, I knew this is what I want to capture. So I went that global feature. But for some proteins, I'm interested in covering side chain movement. So there I won't do something like that. I will use some more local features. So it's a bit human intuition yet, and that's how it is.
Amazing. Thank you so much, Pratyush. Let's get one more round of applause.